Hey guys, so in this video we are going to talk about the cell cycle, and we're going to begin to talk about cell cycle by looking at the structure of DNA, and then we're going to move into how does DNA copy itself so that when we make new cells we end up with multiple copies of the exact same identical strands of DNA. So first, let's review the structure of DNA. Now, we talked a little bit about this at the beginning of the year when we did biomolecules, but it's super important that we review that information because you have to understand how a DNA strand is built. So, our driving questions for this set of notes are: what are the components of DNA? What is the structure of DNA? And number 3, what is the importance of the order of the nucleotide bases in DNA? So, starting with the components of DNA. Recall from Unit 2 that DNA is a nucleic acid. And remember that the monomers of nucleic acids are called nucleotides. So DNA is a polymer built of monomers called nucleotides. And we learned that a nucleotide is made up of a phosphate group, a sugar, and a nitrogen base. DNA stands for deoxyribonucleic acid. So the name comes from the fact that DNA is a nucleic acid, and the sugar that we find in those nucleotides is deoxyribose sugar. So the name deoxyribo tells you the sugar, nucleic acid tells you that it's a nucleic acid. Within that nucleotide, not only do we have the phosphate and the deoxyribose sugar, but we also have 4 different bases that make us 4 different nucleotides. So the 4 bases that exist in DNA nucleotides are adenine, which we represent with an A, thymine, which we represent with a T, guanine, represented by a G, and cytosine, represented by a C. So you can see in each of these pictures, they are part of an entire nucleotide. So we have a phosphate group, the deoxyribose sugar that's a pentagon, and then we have the nitrogen or nitrogenous base, A, T, G, or C. Now, Chargaff was a scientist who studied how DNA bases fit together. And what Chargaff determined is that the percentages of the amount of each of the letters are similar to each other. So whenever he looked at A's, let's say he saw 30 or 39% adenine, he would also notice that there was about 39% thymine. Or if he saw about 10% guanine, there might be about 12% cytosine. So the numbers were always really, really close. And what that told Chargaff is that, well, since the percentages of A and T are very similar, A and T must always be together. And since the percentages of G and C are very close, G and C must always be together. So therefore, Chargaff determined that adenine always pairs with thymine and guanine always pairs with cytosine. A good way to remember this is with the mnemonic device, apples are on the tree and cars are in the garage. Apples for adenine, tree for thymine, apples in the tree, and then C, cars, cytosine, G, garage, guanine. So cars are in the garage. Cytosine goes with guanine. So here's an example of the base pairings, and you'll notice that in between each of these nitrogen bases are some bonds. We call them H bonds for short, but that stands for hydrogen bonds. So the base pairs are held together with hydrogen bonds. G and C have 3 hydrogen bonds between them, and A and T have 2 hydrogen bonds holding them together. These nitrogen bases, along with their corresponding bonds, are what makes up the rungs of the DNA ladder. The sides of the DNA ladder are going to be made up of repeating phosphate, sugar, phosphate, sugar. So let's put the DNA together and look at its overall structure. So just a brief history of DNA. We discovered DNA in the 1860s, and from that time forward, scientists continued to investigate this new molecule, ultimately determining that DNA is what codes for our proteins that give us the traits that we have. Now, through all this time, scientists knew the pieces and parts of DNA. They knew about A and T, they knew about G and C. Chargraph even told us A pairs with T and G pairs with C. But no one really understood the entirety of how DNA fit together. It was like a jigsaw puzzle that no one could assemble. Well, in the early 1950s, two groups of people in two different research labs began to work on the problem. There was James Watson and Francis Crick working in a research lab in Cambridge, and there was Rosalind Franklin and Maurice Wilkins working in a research lab in London, England. And what happened was Rosalind Franklin was taking X-ray crystallography pictures of DNA. And Watson and Crick managed to get a hold of one of these pictures without Franklin's permission, but that's a whole nother story for a different day. Watson and Crick were able to use that picture, photo 51 that you see here on the screen, to determine that DNA must be a double helix. So they cut out some paper models of the parts of DNA and started assembling those. Now, you're looking at Franklin's picture probably, and you're going, well, this doesn't look like any double helix or picture of DNA I've ever seen. That always looks like a little squiggly or a twisty ladder. Well, that's true. A double helix does look like a twisty ladder. But the pictures you've always seen show DNA from the side view, kind of like I'm showing you a side view of this pen. But Rosalind Franklin's picture was taken from the top down. So she was taking the picture looking from the top of DNA towards the bottom, not at a side view. The little X in the middle told Watson and Crick that that was why it was a double helix. So DNA is a double helix consisting of your bases in the middle making up the rungs of the ladder, A, T, G, and C, and the outside is what we call the sugar phosphate backbone. So here's another view of the DNA double helix. This one is drawn more true to the chemical formula. So you can see here's our phosphate, our sugar, and our nitrogen base. So this is one single nucleotide right here. Here's another one, phosphate, sugar, and thymine. And we've assembled those nitrogen bases together, bonding so the nucleotides are combined to make up the DNA double helix. And again, here's that sugar phosphate backbone. So we have these A's and T's and G's and C's, but what's the importance of them? Why do we care what order the A's, T's, G's, and C's go in? Well, if we look at everything that exists on the planet, you'll notice lots of biodiversity. Biodiversity are all the different types of organisms that exist, all the differences in those organisms. Genetic diversity is how the genes that are coding for those traits that give us biodiversity are put together. So, for example, in these Labradors right here, you can see that one of them is blonde and the other is black. The reason why their fur are just two different colors is because their genes are put together slightly differently, which codes for slightly different proteins, which gives them slightly different traits. So the specific order that the DNA bases are put together in determines exactly what protein is made. Change the order of the bases, change the protein that's made, therefore change the trait. So you can see this top strand of DNA and this bottom strand of DNA are put together in two different orders, so they're coding for two different proteins. So that means that all of the biodiversity on Earth can literally be traced back to the differences in the order of the nucleotide bases. So you'll notice here we have some different mammals, and you can see that a human and a chimpanzee have a couple of differences in their order of the bases. So right here, there's an A that's different from the chimpanzee's C. There's a couple other differences. This is just a small piece of the DNA. You can see, though, as we get farther and farther away from humans, like if we look at a mouse, there's some other variations there. And the variations continue the less related you are to an organism. So the more related, fewer differences, less related, more differences. All right, so that's it for this set of notes. In the next set of notes, we'll get into DNA replication. Thanks for watching, guys.